This is Chapter 7, the control of microbial growth. So starting out with some vocabulary and terminology of microbial control, looking at sepsis and asepsis. So sepsis refers to a bacterial contamination. So then asepsis, A meaning without, is the absence of significant contamination. So just like in lab, we practice our aseptic technique. So we don't have any cross-contamination during our lab experiments. Aseptic surgery techniques are also used to help prevent microbial contamination of open wounds in the operating room. Sterilization refers to removing and destroying all microbial life. So it completely destroys all forms of microbial life on a surface. Commercial sterilization to make sure we kill those botulinum endospores in canned goods, so botulism bacteria. Disinfection involves destroying harmful microorganisms, but not necessarily all the microbial life on the surface. Antisepsis is destroying harmful microbes from living tissue, so basically disinfecting a living tissue. So like an antiseptic mouthwash, destroys the harmful cavity-causing bacteria from the inside of your mouth. Degerming is the mechanical or physical removal of microbes from a limited area. So an example would be just washing your hands. So that is an example of degerming. So you have to rub your hands physically together and scrub them with the soap to remove those microbes. Sanitation involves lowering microbial counts to safe public health levels and minimizing disease transfer from person to person. So like in lab, when you have to sanitize your work area at the beginning and end of each lab, so we're applying a sanitizer of approved use, and we're applying physical mechanical force when we wipe the surface. Biocides or germicides are treatments that completely kill microbes. Bacteriostasis or bacteriostatic treatments don't kill the microbes, but they inhibit them, or stasis meaning kind of stable, so it kind of freezes them in place so they can no longer multiply. Bactericidal is homicidal toward bacteria, so it's going to kill those organisms, whereas bacteriostatic just prevents them from multiplying. So if the chemical agent or antimicrobial agent is removed, these bacteria could potentially start reproducing again. So this is showing the effectiveness of different types of antimicrobial treatments. So we have a lawn growth of bacteria on our Petri plate, and we look for the zones of inhibition. So these are areas where there's no bacterial growth. So this is showing that chlorine seems to be the most effective on these different bacteria. And it also shows that different antimicrobial agents have different levels of effectiveness against different types of microorganisms. So in the previous chapter, we talked about exponential growth of microbial colonies. When microbes die, they also die at an exponential death rate. So this is typically graphed on a logarithmic scale. So we have that nice straight line. So if we apply a cleaner that has a death rate of 90% every 1 minute, so every minute 90% of the population is killed. So if we were to graph this arithmetically, it wouldn't be a very feasible graph. So the log scale just gives us a nice even line. So we start with a population of 1 million cells. After a minute, 90% has been killed. We're left with 100,000. Another minute passes. We're down to 10,000. 90% of those 10,000 die. We're left with 1,000. 90% of those die. 100. 10, and then finally 1. So this particular example, our cleaner takes 6 minutes to effectively kill 99.9% of the bacteria. So this is why it's important to make sure you read the label on your household cleaners to make sure you're leaving the antimicrobial substance on the surface long enough to reach that 99.9%. So in this example, if we only left it for 2 minutes, before we wiped it off the surface or cleaned it up, we would still have 10,000 of those bacterial cells remaining. The effectiveness of our treatments are going to depend on a few different factors. So one being the number of microbes. So the higher your population load, right, the more cells you have to kill, the longer it's going to take, right, to reach that 99.9% effectiveness. Right, so this example, we start with a lower population, right, so they're all completely killed within 3 minutes. Right, but if we start with a higher population load, it's going to take more time. The environment can also influence the effectiveness of your treatment. So presence of organic matter, how it reacts with the cleaner, temperature, presence of biofilms. Remember, the biofilms can be a little bit more resistant to antimicrobial agents because they have those layers of bacteria. So the cells on the surface would be affected, but the cells in the lower layers would be protected. Time of exposure, like we said, so make sure you read the labels and make sure you're leaving your cleaners on long enough for them to be fully effective. Right? So if we were to remove our cleaner after 3 minutes, right, we still would have a relatively high population remaining. Effectiveness can also depend on microbial characteristics. So different types of cleaners have different levels of effectiveness on different microbes depending on their own characteristics like cell wall structure or metabolism. So there are 3 main mechanisms of action or plans of attack that these antimicrobial agents can take when they are killing these microbial cells. So one action could be to alter or damage the cell membrane. So the cell membrane is kind of like the skin of the cell. So if we poke holes in that membrane, now the internal cell contents can leak out and other harmful substances can now get in the cell. Some microbial control agents may target the proteins or enzymes of the cell. So remember, enzymes pretty much control and regulate all cells' metabolic activities and those biochemical reactions. And enzymes are just proteins. And everything goes back to structure reflecting function. So if we denature and change the shape of those proteins, they're no longer functioning. So those cells can no longer undergo their usual metabolism. Another angle of attack would be to damage the nucleic acid, so the DNA or the RNA. So the DNA is kind of the base source of information on how to make these proteins and enzymes and how to assemble and make new cells. So if we damage the genetic blueprint or the instructions, then the cell will no longer be able to make the proper proteins and undergo its usual metabolism and growth. There are 2 main categories of methods we can use to control microbial growth, physical or chemical. So one of the most basic types of physical methods would be heat, because heat is going to denature those enzymes, right, and essentially make the cell unable to undergo its metabolic reactions. When we look at a thermal death point, we're talking about the lowest temperature at which all the cells in a liquid culture are killed in 10 minutes. Right? So 10 minutes is kind of our baseline. Right? So what temperature does it have to be for all the cells to be killed in that 10-minute time frame? So this figure is showing that most all of the pathogenic bacteria and harmful insects in this particular soil will die between the 60 and 80 degrees Celsius range in about 10 minutes. So this would be their thermal death point. Thermal death time is the minimal time for all the bacteria in a liquid culture to be killed at a particular temperature. So instead of presetting the time, we're presetting the temperature and then saying how long it takes at this temperature to kill all of those cells. Right, so in this particular example, we're starting at 0, right, and by 45 minutes, right, we've killed all of the cells in the population. Decimal reduction time, or DRT, is the minutes it takes to kill 90% of the specific population at a given temperature. So related to the thermal death time, but we're only looking at 90% of the population killed. We can also use moist heat sterilization for microbial control. Moist heat works by coagulating and denaturing those proteins. So boiling is one method we could use to sterilize surfaces and objects. We can also use free-flowing steam. So if steam's hot enough for the water to evaporate, those high temperatures can denature the proteins of any microbes. In lab, to sterilize our lab equipment and media, we use an autoclave, which utilizes steam under pressure. So it's about 121 degrees Celsius at 15 pounds pressure per square inch for 15 minutes. So that high pressure steam kind of washes over our objects we put in there and sterilizes them. Moist heat sterilization is good for killing all organisms and endospores. So not all methods will kill endospores as easily. But the steam has to contact the item's surface for it to be effective. The larger a surface area an object has, is the longer sterilization time it will require. So again, we said the steam has to make contact with the surface of the object. So if we have more surface to cover, right, it's just going to require more time to make sure everything makes contact. So this example showing just your standard test tube, right, we can sterilize in about 15 minutes. But a larger flask or bottle, right, in the thousands of milliliter range, right, could take half an hour or more, right, to reach that full sterilization. Sometimes we can use test strips to indicate if the objects have been sterilized. So if we left it in the autoclave long enough, we'll have our indicators, or we'll know if we need to put it back in for a little while longer. But also keep in mind, not all objects can withstand moist heat sterilization. So some things will melt under those high temperature and pressure conditions. So remember, Louis Pasteur developed the process of pasteurization to reduce spoilage organisms and pathogens in beverages. Right, so we still use this process today. Right, so it utilizes high temperature for a short time, so 72 degrees Celsius for 15 seconds. So it's not necessarily long enough and hot enough to kill and break down all of the cells. Right? So we don't want to change the taste and overall texture of our food product, or in this example, our milk. We apply just enough heat to denature the enzymes of the bad microbes. Right? We don't want to spoil our milk for that 15 seconds, and then we rapidly switch it over to a cooling phase. So then those enzymes are kind of frozen back in that denatured state, right, and ultimately unable to function. However, it doesn't kill all of the microbes in the product. So some organisms still survive. These thermoduric are able to survive those high temperatures. So this is why, even though a lot of food items are pasteurized, they don't have an indefinite shelf life. There's still some slow growing spoilage bacteria in those. Dry heat sterilization kills by oxidation. So this is essentially just an open flame. So like a Bunsen burner or incinerators in lab, right, when you flame your loop, you're just incinerating and burning any microbes on that surface. Hot air sterilization is basically like a sterilization oven. Right? So, but still using the same principle of dry heat sterilization. So like we said, not all substances can withstand those high heat and temperature methods. So another method we could use, a physical method, is filtration. So passage of a substance through a screen-like material. So this will be used for those heat-sensitive items. HEPA filters are very efficient at removing microbes and particulate air as small as 3 micrometers. So HEPA stands for high efficiency particulate air filters. If we need to filter even smaller microorganisms like viruses or proteins, we can use membrane filters. So any microbe or particle, right, would get trapped in this filter, and then we're left with our sterile filtrate. So just as heat was a physical method of microbial control, low temperatures and freezing can be another method. However, whereas heat had more of a bactericidal or bacterial killing effect, low temperature is primarily a bacteriostatic effect. So your standard refrigeration, deep freezing, and freeze drying. So even though we put our food in the refrigerator, it still will go bad in a few days. It's not going to last indefinitely in the fridge because of those psychrophiles, or those psychobacteria that like to live in those cold conditions. And again, it's primarily bacteriostatic because if we take the food out of the fridge and it's left at room temperature, those bacteria will start to multiply again. Desiccation is the absence of water. So all living cells require water. Right? So without water, they cannot undergo their metabolism, right, or grow and divide. But again, this is more of a bacteriostatic effect because once water is re-added, right, so once we rehydrate that substance, then the microbes can continue to grow. So freeze drying is just kind of an extreme form of desiccation. So things like coffee, some meats and vegetables. Osmotic pressure can also be a physical method of control by using high concentrations of salts and sugars to create those hypertonic environments. So remember, hypertonic is where the solution is more concentrated than inside of the cell. So water via osmosis always wants to go where the solution is more concentrated because it wants to dilute it out. So water will be drawn out of the cell into that solution and causing plasmolysis. So things like jams and jellies, beef jerky, have longer shelf lives because of those high osmotic pressures. However, there are some of those halophiles, like Staphylococcus aureus, that can still grow in these high salt conditions. Radiation can also be used to control microbial growth. So ionizing and non-ionizing radiation, pretty much everything below the visible spectrum, can work to control microbial growth by damaging DNA, by causing mutations. Microwaves can also kill some microbes, but it's more so by heat, by generating heat in the food, not so much from the radiation itself and attacking the DNA structure. Now looking at some chemical methods of microbial control and effective disinfection. So one important component of the effectiveness of your disinfectant would be the concentration. So again, it's important to read the label on your disinfectant bottle. Different disinfectants work better at different concentrations. So you might have to do 2 parts water to 1 part disinfectant, or 3 parts water, depending on what it is. So make sure you follow all the label directions. The presence of organic matter can also influence the effectiveness of a disinfectant. So, for example, with chlorine, right, if there's certain organic matter present, it can react with that as well and cause some carcinogenic byproducts. pH is important in the effectiveness of a disinfectant. So in this example, chlorine is most effective between 5.5 and 7.5 pH. So this is important if you have a pool, right? You also have to maintain certain pH level or your chlorine won't be effective at keeping your pool clean. And then finally, time. Time is another important factor in your disinfectant's effectiveness. So remember, read the label and keep the surface wet for that required contact time. So remember, if it kills 99.9%, but its decimal reduction time, right, to kill 90% of the population is 1 minute. Right? We had that example with the million cells that took 6 minutes to kill 99.9%. Right? So we would have to make sure our surface stayed wet that entire 6 minutes. If we wiped it up or cleaned it up before that 6 minutes, we would leave a lot of bacterial cells behind. Right? So in order for them to work and be most effective, make sure you're following that contact time. The Disk-Diffusion Method evaluates the efficacy of different chemical agents. So your filter paper would be soaked in different chemicals, right? Or we could also do this with antibiotics. And we're just going to look for that zone of inhibition, or that clearing around the discs. So this means there are no bacterial cells growing or present, right? Because the chlorine has killed them or prevented them from growing and multiplying. And again, showing that different chemical agents can have different levels of effectiveness against different types of organisms. So a quat disinfectant has moderate effectiveness against Staph aureus, but not really any against Pseudomonas. Phenols and phenolics operate by injuring the lipids of that plasma membrane. Right? So we're going to attack the membrane. Remember we said there were three basic plans of attack, either the membrane, the proteins and enzymes, or the DNA. So these specialize in attacking the membrane, essentially causing all the cell contents to leak out. Bisphenols are essentially just two phenols linked together. A common one found in your everyday antibacterial hand soaps is triclosan. And these also work by disrupting and attacking those plasma membranes. Chlorhexidine is a biguanide that's used in surgical hand scrubs. It also attacks the cell by disrupting the plasma membranes. Halogens are everything in column 7 on the periodic table. Iodine is commonly used for microbial control as either a tincture or an iodophore. So tinctures are just the iodine solution mixed with alcohol. Iodophores are iodine combined with organic molecules. But they are all going to work by impairing the protein synthesis and altering the membrane. So it could attack the RNA or the DNA of the cell, so it's not able to make those proteins and enzymes to carry out those metabolic reactions. So betadine is a commonly used one, used to clean skin surfaces before surgical procedures. Chlorine is an oxidizing agent that's going to shut down those cellular enzyme systems, essentially by attacking the chemical bonds in those enzyme proteins, right? Ultimately denaturing the protein and causing it to lose its function. Alcohols are used for disinfectants and antiseptics. They work by denaturing proteins and dissolving the lipid membrane. They're very effective at killing bacteria and fungi. However, they don't really have an effect on endospores or those non-enveloped viruses. So an envelope is just basically kind of like a cell membrane. So it's a lipid membrane around the viruses. So if it dissolves lipids, but there are no lipids on these viruses, then it's not going to be very effective. Viruses that do have envelopes, like coronavirus, is a enveloped virus. So alcohols, like found in hand sanitizer, would be effective at killing those. Ethanol and isopropanol are the most commonly used, and they're most effective at 70%. So remember we said concentration of the disinfectant was also important in its effectiveness. So alcohols work kind of as a two-prong approach. So we attack the membrane lipids, we dissolve those lipid membranes, and we denature and destroy their proteins. Heavy metals and their compounds work by oligodynamic action. So it only really takes very small amounts of these heavy metals to have an antimicrobial effect. The most commonly used ones are silver, mercury, copper, and zinc. So we use silver nitrate on newborn babies, those eye drops, to help prevent ophthalmia neonatorum, which is basically conjunctivitis in newborns. We have mercury additives in paint, right, to prevent mildew and microbial growth in paints. Copper sulfate used in algicide for pools, so pool chemicals. If you have a pool, if you ever see the word blue, like on the label for the pool chemical, that means it has copper in it. Zinc chloride found in mouthwash. So this is showing those zones of inhibition or lack of bacterial growth where these heavy metal coins are making contact with the agar surface. And fun fact, brass doorknobs automatically disinfect themselves in about 8 hours. Surface active agents, sometimes referred as surfactants or just detergents. Right? So they're going to work on the surface of an object to remove microbes. So things like soap work by degerming and emulsifying the microbes and oil globules. Acid anionic sanitizers contain anions, or those negatively charged ions that react and disrupt the plasma membrane. Quaternary ammonium compounds, sometimes called quats, are cations, right, those positively charged ions that can actually kill the cells. So they denature the proteins and disrupt the plasma membrane. So a standard type of soap or surfactant agent would be like just your Dawn dish soap, right? So, or any hand soap really. They all work about the same. Right? So they work as surfactant, so they're lipid-based. Right? So remember, lipids dissolve lipids, like dissolves like. Right? So we have this lipid-based soap that's going to emulsify or form these fat or oil globules. Right? So these are basically like little, think of phospholipids. Right? So we have a hydrophilic head and a hydrophobic tail. So the tails will make contact with in this example, a stain, or just say an oil globule, or with some microbes on it, or some dirt on your hand. So they'll surround the oil on your skin, right, with the microbes, but it's not going to detach on its own, right? So we have to have that mechanical agitation. You have to rub your hands together, right, for at least 20 seconds, right, to lift all of that oil and microbes off the surface and allow these surfactants to form these globules. Right? And once they're lifted from the surface, we can then wash them away with the water. So comparing the effectiveness of various antiseptics. So different types of antimicrobial agents will have their own kind of specialization or planned mechanism of action. And different disinfectants will have different levels of effectiveness against different types of microorganisms. So your biguanides, phenolics, and your quats primarily target the plasma membrane. Some aldehydes, radiation, we said targeted the DNA, cause mutations. Halogens, metals, phenolics, alcohols can also target the proteins, those enzymes. So this is showing a percentage of particular type of bacteria surviving different treatments. Right? So with just soap and water, right, we didn't really remove that many microbes at all. Right? So the most effective was our iodine with 70% ethanol. So because our modern lifestyles have given rise to high levels of food production, right, more mouths to feed, and a greater demand for food shelf life, we have also increased use of food additives, these chemical food preservatives. So these are things to help preserve flavor or improve taste and appearance in a food, as well as the shelf life. The most commonly used are your benzoates and nitrites. Sulfur dioxide can prevent wine spoilage, so the spoilage bacteria in wine. So the benzoates, benzoic acid, nitrates work to prevent endospore germination. Antibiotics are proteins that are produced by one microorganism that inhibit the growth of another. We use some antibiotics to prevent spoilage of dairy products, as well as livestock and meat production. So we can see the effectiveness of different antibiotics on different bacteria by using those zone of inhibition plates. So we'll do this in lab later in the semester just to see which bacteria or which antibiotics are most effective against certain bacteria. Aldehydes work by inactivating those proteins by denaturing them. So this is used for preserving specimens and in medical equipment. So things like formaldehyde that we use to preserve our dissection specimens, our specimens for dissection. So different type of microbes have different degrees of resistance to our standard antimicrobial treatments. So some of the least resistant are your viruses with lipid envelopes, your gram-positive bacteria. Some of the most resistant are the endospores, the mycobacteria, right, that have those thick outer layers in their cell walls, and prions. We'll talk about prions later, but prions aren't really cells, they're just proteins. So a lot of our antimicrobial agents target, either the cell membrane or the DNA. Well, prions don't have either of those things. So this table is showing the effectiveness of different types of chemical agents against these highly resistant endospores and mycobacteria. You'll see not all antimicrobial treatments are effective for all bacteria. So alcohols were pretty poor against endospores, but relatively good against mycobacteria, whereas chlorines were fair against both. So again, just showing that different types of antimicrobial agents have different levels of effectiveness against different types of microorganisms, depending on their own individual characteristics.